Chilodonella is a ciliate, a member of the phylum Ciliophora. It resides in its own named genus, Chilodonella. In the genus Chilodonella, there are many named species. Over 40 are listed in Call's textbook. Chilodonella can reproduce either asexually or sexually. As is the case with many ciliates, Chilodonella has two nuclei, a macronucleus and a micronucleus, and each of these undergoes a different type of replicative process during asexual reproduction. During asexual division in Chilodonella, the germline nucleus, the micronucleus, undergoes mitosis, while the somatic nucleus undergoes amitosis. Parenthetically, amitosis is a process of cell division in which there is no mitotic spindle formation, and the duplicated chromosomes of the somatic nucleus are simply separated by binary fission. Overall, this process produces two daughter cells which have identical germline nuclei, but potentially different somatic nuclei. One of the first easily recognizable signals of impending cell division in Chilodonella is a crease in the pellicle of the organism at mid-body. While the development of this crease is progressing, the organism seems to behave normally, despite having undergone major changes in its DNA inventory. It should be noted that the anticipated anterior daughter cell retains its capital structures, while the more distal posterior daughter cell will have to form new ones, a process which will not be completed until the two daughter cells are close to total separation. This video shows late-stage division in Chilodonella. The anterior daughter cell has retained its fundamental structures, most notably its capital structures, while the posterior daughter cell is in the process of relatively quickly forming them. There does seem to be an element of the birthing struggle involved here, with seemingly agitated random movements. However, it is clear that these movements facilitate both the drawing out of the ever-thinning cytoplasmic connection between the two daughter cells and the final breakage of this connection, which will result in the release of two new organisms. Briefly, in this portion of the video, the active capital cilia of the anterior daughter cell can be seen. No doubt they are assisting in the generation of the conjoined organism's circular movements. We are reminded by this video that the asexual division of ciliates is a transverse process. Note that the process of division is more or less a co-equal one, and that the size of the two daughter cells will be approximately the same. At this point, The developing capital area of the posterior daughter cell can be seen, hanging on literally by a thread now. Finally separated, functioning independently, perhaps a little disoriented and agitated by their newfound freedom.